bit more background around ALS and research.
Hi, everyone. I'm happy to know most of the people in the room. And I just wanted to say a few words to add on to what Sarah said, is that our chapter of the ALS Association here in Oregon and Southwest Washington, one of the primary things that we do is we try to connect with families and help them live the fullest life you can with ALS. And that's a big primary focus. But we have a dual mission. Our other focus is to work to create a world with ALS. And so funding ALS research is part of that charter. And as one of 42 chapters of the ALS Association across the United States, we all contribute globally to the Treat ALS research program through the National ALS Association. And it's a multifaceted program that supports research grants that go from everything from basic science research about motor neurons and functioning and basic physiology of ALS, to translational research, how things get transferred from the clinic, from the lab bench into the clinic, as well as supporting targeted clinical trials that may not get footing somewhere else to help get things started in looking at human applications, as well as clinical management research, aspects around what are the supports and resources that help families cope with ALS and live a good life with the disease. So all of this together, I'm proud to say that currently right now the ALS Association is funding over $20 million in active research grants, and our chapter is part of funding that. Each quarter we send funds to our national organization to fund research, but I'm really happy to share that over the last 3 years, on top of sort of what you call our regular contribution, because of the success of things like our Walk to Defeat ALS, we've been able to send over $350,000 of additional monies to go just directly to fund ALS research programs. So, and that's because of all of you in this room who care about ALS. So, with that being said, I'd really would like to just, glad so many people here are passionate about ALS research. You know, your support of our chapter is a commitment to research, and it's a commitment to helping families here in Oregon and Southwest Washington. So thank you very much, and I'm so happy you're all here.
Okay, and before we invite up Dr. Goslin, our first presenter, I want to give you a little overview of what today will look like so that you know, most importantly, when to take your breaks when we've got that planned. So we will have Dr. Goslin up here for close to an hour, as much time as it takes, 45 minutes to an hour. So around 2:30, we'll break for about 15 minutes, and there will be refreshments, some new ones out there, so feel free to go and get some of that and use the restroom, which is down the hall if you haven't seen it, in the back behind the ramp. And so then we'll come together again for two presenters. We'll have Dr. Beckman and Dr. Morton give their presentations all together. That will be another hour or so again. We'll take another 15-minute break. That gets us to about 4 o'clock, when we'll come back together for a panel discussion of questions and answers. So kind of as presenters, you can say whether you'd like some questions interactively during your presentation, but we will have 30 minutes dedicated to questions and answers for all three presenters at the end. So we are honored to have today with us Dr. Kimberly Goslin from the Providence ALS Center. Dr. Goslin is an MD, PhD, and graduate of University of Virginia. She did her neuromuscular fellowship at, I might not say this 100% correctly, but Brigham's and Women's Hospital at Mass General, and she founded the ALS Center in 2005. So those are some things on paper about Dr. Goslin, but as everybody who has met her knows, it's an honor to work with Dr. Goslin one-on-one. Her brain and her heart battle each other for size, and today we'll get to see a little bit about what the work that she's done and how she will present ALS research to us. So with no further ado, Dr. Kim Goslin.
All right. Can everyone hear me? Is that adequate? It's nice to look out and see so many friendly faces. It makes it a little easier to talk. So I've been asked to give an overview of what's been happening in the field of ALS for the last couple of years and what's coming up. And it's been a huge year in ALS in terms of breakthroughs and discoveries. You can't hear? it's a huge year in ALS in terms of breakthroughs and discoveries. And I come out of research conferences feeling very hopeful about what's going on in ALS. And I hope I can fully convey that to you, because it's really quite striking what's going on. And so the main message of this is hope. So I know most of you know quite a bit about ALS, but I just wanted to make sure we're all at the same level, that we understand the basics. And then I'm going to talk about recent research findings in genetics, pathogenesis, and stem cells, and then ongoing and future clinical research. Just give you a sampling of what's going on. So the basic is that ALS is a motor neuron disease. And I think you all know what happens. Motor neurons die, and because of that, muscles waste away. And people develop gradual paralysis of all voluntary muscles. So muscles of the arms, legs, speech, and swallowing. Not the heart muscle or bowel or bladder. And this, just the anatomy of the motor neuron, it has dendrites and then this long axon that goes out to the muscle. And that axon can be several feet long as it goes down to the leg or to the arm to reach its muscle. And I've mentioned here, ALS is noted for having a segmental spread of disease. And that'll be very important in terms of some of the research that's being done. ALS generally starts in an area and spreads contiguously along the nervous system. So it might start in the hand and spread up the arm and then to the bulbar muscles. And there's been very important discoveries about what underlies that in terms of the pathogenesis. So I'll come back to that in a little bit. So ALS is very unique in that it affects what are called upper and lower motor neurons. And when we talk about upper and lower, we're not talking about arms versus legs. We're talking about the connectivity of the nervous system. And so the upper motor neuron comes from the brain and travels down the brainstem and spinal cord. And then that connects to the lower motor neuron that goes directly out to the muscle. And when lower motor neurons are affected, you get wasting of muscles, cramping, twitching, fasciculations, and weakness. And when the upper motor neurons are affected, you get spasticity, hyperactive reflexes, stiffness of the muscles. And there's very few things that give you that unique combination of findings that lead to the diagnosis of ALS. Just some basic facts. The incidence of ALS is about 2 to 3 per 100,000. The average age of onset is about 54 to 56. It actually increases as people get older, and then after about 70, the incidence starts decreasing. And it's slightly more common in men than female. Average life expectancy is 3 to 5 years, but it ranges from 6 months to 20 years. And notably, people with ALS progress in a linear fashion. They don't tend to speed up or slow down. 5 to 10% is familial, and there's an increased incidence in veterans, people who've had head trauma or pesticide exposure, and athletes, notably soccer players, and smokers as well. So I've just spent the last 5 minutes saying how ALS is a motor neuron disease. But some of the big breakthroughs that have come in the last few years is understanding that it's not just a motor neuron disease, that it affects many other types of cells. For instance, we know that frontotemporal dementia can be associated with ALS, and those are affecting neurons in the frontal lobes. But it also affects other types of neuronal cells called glial cells. And the glial cells are the nursemaids of the neurons. They support them and help keep them healthy. And so I just put this slide up because it's pretty. I used to do a lot of basic research, and I always loved looking at the pretty slides. But so this is a motor neuron, and it has that long axon. And then in red are the astrocytes, or the glial cells. And they interconnect with the neurons and support them. And as it turns out, they play a very big role in ALS. And I'll come back to that as well. So there are many factors. So one of the things about ALS is it's a syndrome that has many causes that lead to the same final pathway of motor neurons dying. And there are many factors that are involved in the motor neurons dying. And this is just a list of some of the things that have been talked about, and like glutamate toxicity, mitochondrial dysfunction, inflammation. And I've highlighted the two that are actually becoming most important in terms of research. Abnormal protein aggregation seems to underlie a lot of ALS, and RNA dysfunction is also a significant part. And these discoveries have related to some of the new genes that have been identified as causes of ALS. And at the end of the talk, I'm going to be going through some of the clinical trials, and I want you to keep this slide in mind because all of these issues are targets for medication to treat ALS. So in the last 2 years or so, there's been an explosion in the genetics of ALS. And the first gene for ALS was discovered in 1993, and it was the gene for superoxide dismutase. And subsequently, in the last 2 years, about 33 other genes have been identified. So it's a major discoveries. And so now, when we have someone with familial ALS, we can often or usually identify what the genetic cause of that is. So 75% of cases. And I can't emphasize enough how these genetic discoveries have led to the development of new models, very encouraging models, and new avenues for studying ALS and developing treatments. This is just a partial list of some of the genes that cause ALS. And ALS, SOD1, like I said, was the first one. And it's the one that all of the models of ALS have been developed have been used up until recently. So they have a mouse model that develops ALS based on this genetic mutation. And all of the research has been done on this model with very inconsistent results. And TDP43, Dr. Morton, I believe, is going to talk about. And C9orf72 is one of the newest genes that's been identified and actually accounts for about 40% of all cases of familial ALS. So it's the most common. It also accounts for about 10 to 15% of sporadic ALS, so people that don't have a family history. And so it's a very important discovery. This gene is also highly associated with frontotemporal dementia as well. So when we look at all of the genes that cause ALS, there's some themes in terms of what they do. So I think probably the most important that I'm going to be talking about more is disturbance of protein stability, a tendency for abnormal proteins to aggregate, and by aggregating, causing damage to the motor neurons and cell death. Disturbance of RNA binding proteins is also very important, and disturbance of cytoskeletal function. The cytoskeleton is basically the backbone of the neuron, and when that doesn't work, the neuron deteriorates. And some genes also cause susceptibility to environmental toxins. So it is now recognized that familial ALS is likely underestimated, and that and the reason for this is that in modern society we tend to have small families, and some of the genes that cause ALS have what's called incomplete penetrance. So you can have the gene and still not develop ALS. Because of that, it's thought that sort of genetic causes of ALS are more common than we've estimated. And so the approach to looking at ALS now, instead of talking about sporadic versus familial, we talk more about a burden of disease-causing factors. And so you have to get to a certain level or threshold of a burden before you manifest ALS. And so what makes up that burden is genetic load, so genes that cause ALS and genes that prevent ALS. And then cell damage that occurs naturally as people age, and then environmental factors. So you can get all sorts of combination of things. You can have a gene that always causes ALS, and so you get ALS. You can have a gene that makes you slightly more likely to have ALS, and then you can have a head trauma, and so you might be more likely to get it. So it's a combination of factors, and there are probably many factors that aren't, strictly speaking, single genes that are passed down that may make you more likely genetically to get ALS. And so one of the important concepts is that there are genetic modifiers of disease. So one of the things that's well known is that when in a family that has a certain mutation, that family members can progress very differently. Some progress rapidly, some start in the bulbar area, some start in the hand. And it turns out that there are factors that affect that and influence the progression of ALS. So one of these is a protein called ephrin B2. And it's a protein that prevents damaged motor neurons from being repaired. And it's actually at increased levels in people with ALS. But what's been shown is that in people who have ALS, if they have a particular mutation of this gene for this protein that results in lower levels of the protein, then they progress more slowly. And so there are a number of modifiers like this that are being studied. And there are already in development ephrin B2 blockers that are being developed to alter the progression of ALS. So it's a very exciting area. It's funny how your throat always gets a little dry when you're up here talking. ALS pathology. And so what this is a picture of the cell body of a motor neuron. And the big arrow is pointing to this abnormal accumulation of stuff. And those used to be, and this is the hallmark of ALS pathology. And these used to be called Burnina bodies, but now they're just considered aggregated protein. And in the last few years, we've learned more and more what those are. And it turns out that a significant portion of this aggregation is made up of a protein called TDP43. And so because this is the common pathology that people with ALS have, most cases of ALS are considered TDP43 proteinopathies. And the reason this is important is that the model that has been present to date, which I mentioned earlier, most of the animal models are based on SOD1. And it's notable that the SOD1 models don't have that abnormal accumulation of TDP43. So it may be that this model that we've been using may not be the best model for ALS. And that's really important because so many of the studies that have been done in SOD1 mouse that looked exciting, haven't translated into humans. And so this is just saying that now we have all sorts of new models based on the new genetics. And it's very hopeful for developing new understanding and new treatments. So I think this is one of the most interesting things that's been discovered about ALS recently. And in retrospect, it seems so obvious, but ALS is considered a prion-like disease. And this holds for many neurodegenerative disorders like Alzheimer's and Parkinson's. And you all know, probably the most commonly known prion disease is mad cow disease. And mad cow disease is considered sort of an infection, but ALS is not an infection. So I just want to emphasize that before I go on. But so what happens in prion disease is you have an abnormal prion protein that has an abnormal shape, and it combines with a normal prion protein. They combine, and then the abnormal protein changes the configuration of the normal protein, and so you have two abnormal proteins. So it's proteins that are self-propagating abnormalities. And it turns out that this is what happens in ALS. And so many ALS mutations make proteins that are prone to self-aggregation or glomming together and to changing the shape of the proteins. And that aggregation of protein is part of what's toxic to the motor neurons. So for instance, in SOD1 ALS, superoxide dismutase is an antioxidant, but the mutation doesn't cause ALS by affecting antioxidation. It causes ALS by accumulation of toxic abnormal protein. So what happens is that you have an abnormal protein that makes other proteins abnormal and cause toxicity to the cell. The neuron dies, and then the abnormal protein is taken up by adjacent neurons, and it goes through the same process. So it's thought that this is what underlies the segmental spread of ALS, that ALS is spread through the neuronal connections throughout the nervous system. And so this is particularly exciting because these misfolded aggregated proteins are an excellent target for therapy. And there are already some therapies that are getting into clinical trial to attack the misfolded aggregated proteins. It's not possible, I don't think, to give a ALS update without addressing stem cells. And stem cells in ALS, I think that we tend to think of them incorrectly when we think of what they could do in ALS. And so when I talk to people, it seems like people think that, and you know, stem cells are a type of cell that can become any type of cell. You can make them into any type of cell. And so I think most people think that what's going to happen is that you take a stem cell, you make a motor neuron, you get it into the nervous system, and it develops a connection with the muscle to make the muscle work. And there are just too many challenges for that. That's not likely to happen in the foreseeable future because to get them where they need to go, you have to inject them into an itty-bitty tiny space in the brain and the spinal cord. And then those neurons have to survive in a toxic environment where all the motor neurons around them are dying. And then they have to extend an axon that has to grow 2 to 3 feet to get to its muscle through an environment that is hostile, basically. So that's not the approach that's being used when they talk about stem cells in ALS. There are many alternatives that are being used, and the main focus in ALS is transplanting stem cells that have the ability to make the environment around the motor neurons healthier. And we can do this in a number of ways. One is to make healthy glia, the nursemaids of the neurons, and put them into the spinal cord or the brain, and so that they can detoxify the environment. You can also put in cells that release healthy growth factors, or that modulate the immune system to make it less inflammatory. And cells can also be used as a way of getting new genes and drugs into the central nervous system. So this is the approach that's being used with stem cells. And there are 2 types of human clinical trials that are being done right now. And the first is Brainstorm, and there's no data on this one. But the approach is to use mesenchymal stem cells. So those are stem cells that are taken from a person's own bone marrow or fat cells. And then they are reprogrammed to become glial cells that release growth factors. And then they're reintroduced into the nervous system, either injecting into the spinal fluid or into the muscle. And so this is one approach, and it has benefits in that these stem cells are very plentiful, and it also, because you can get them from your own body, you don't reject them. But like I said, there's no data that I'm aware of with this trial. The other approach is called NeuroStem. And this is where they take stem cells from human fetal spinal cord, and they expand them in culture or in test tubes, and they make them into neuronal cells that have some characteristics of motor neurons and some characteristics of glial cells. And then they inject them into the spinal cord. So it's a direct spinal cord, and the spinal cord is about that big. And so you're injecting hundreds of thousands of cells into a very small area. And so they had to develop new technology to do this. And so there's a new stabilization and injection device that's fairly high tech that allows you to really target that small area of the spinal cord. And so there is preliminary data on this trial so the idea of this is is neuroprotection, again, to make a healthier environment for the motor neurons. And so to date, about 15 subjects have been looked at, so very small numbers. And 12 of them underwent injection into the lower part of the spine, and then an additional 6 received injections into the cervical spine, some getting cervical and lumbar at the same time. And again, because these cells are not coming from the person's own body, these patients need to be on long-term immunosuppression. And the theoretical benefit is neuroprotection. And so there are preliminary results, and they're very exciting, they're very encouraging. So there are some basic things in terms of safety. So one thing is that they found that there didn't seem to be any acceleration of disease progression or major adverse events. And there's a lot of concern about that because the spinal cord is so packed with cells that you could easily cause damage. So they found also that injection of more cells in multiple locations seemed to do better, and that patients who didn't have bulbar involvement also seemed to do better. They're very small numbers, but there are hints of positive effects. And so one of the things they did with these trials is that I've mentioned that ALS tends to progress linearly. So they watch these patients for 3 months or so before injecting so that they could extrapolate what their progression would be. And what they found is that they seem to progress more slowly in a number of variables, particularly between months 1 and 6 after injection, and then it seemed to lose its effect. So very small numbers, but very exciting in terms of being able to show some benefit even with small numbers. So this is obviously moving much more rapidly now in terms of going to the next stages of trial. I think the most exciting stem cell breakthrough is this type of stem cell called induced pluripotent stem cells. And what this is it's the ability to do a skin biopsy on people with ALS and then to turn the fibroblasts, the skin cells, into pluripotent stem cells, so cells that can become anything. And then make them into different types of neuronal tissues and actually study ALS in petri dishes from people who have ALS. So this is a huge technology in terms of advancing our ability to study and treat ALS. I think this is probably one of the biggest breakthroughs in the last couple of years. And so what it allows you to do is to study pathways that you really can't study because you can't get at a human spinal cord or brain, but you can study those pathways. It also allows the ability to very rapidly screen large numbers of potential therapeutic interventions for ALS. And the researchers who work on this postulate that rather than taking 10 years to develop a medication, that you can go from the animal to human in just a couple of years. So I'm going to give you some examples of some of the things that's already been done with these induced pluripotent stem cells. And one of the things that's been done is to make ALS motor neurons in culture and to study the basic electrical currents in those motor neurons. And one of the things they found is that the neurons are hyperexcitable. And when neurons are hyperexcitable, it makes them more prone to damage. And this seems to be because of a deficiency in potassium channels. So immediately, this has resulted in a potential therapy for ALS. There's a medication called retigabine, which is it's an approved medication for treatment of seizures that works on potassium channels to make them work more effectively. And so this may be a potential therapy in part for ALS. And when they looked at motor neurons, ALS motor neurons in culture, they found that this medication corrected that hyperexcitability and the tendency to die. So that's a big breakthrough. This technology has also been one of the pieces of evidence that shows us that ALS is not just a motor neuron disease. And so what they've done is they've taken these iPS cells, they've made them from ALS patients, they've made them into ALS glial cells, and then they've taken normal motor neurons from fetal tissue and combined them. And what they found is that the ALS glial cells cause normal motor neurons to die. So this has led to the concept that, at least in part of what's happening in ALS, is something may be being released by the glial cells that contributes to ALS, to toxicity to motor neurons. And so, again, one of the things I mentioned is that glial cells are much easier to replace than motor neurons. So this is also another area of potential therapeutic intervention. So now what I'm going to switch to is current and future clinical trials. And this is really just not in any particular order. It's just things I found interesting because there's so much out there. And I'm just going to basically just go through this list. The first thing is a protein called NOGO. And I remember when this first came out, I really thought it was fascinating, and I thought it would be a useful, have potential therapeutic interventions for many neuronal diseases. And so NOGO is a muscle protein that inhibits nerve growth. And so you can imagine that its role is that once the nervous system has developed and all of the nerves go to where they need to go, that you don't want them growing anymore. So this protein inhibits that growth. And so it's also a protein that is at higher levels in people with ALS. And so what they've developed is an antibody to the protein that basically destroys the protein. And that's called ozanimab. And this is now in clinical trial. And so what they've done is they've taken this medication and used it on SOD1 mice and found that the motor neuron survival was significantly improved, and the motor neuron force, the muscle force was stronger. So the theory is that in ALS, that this protein, that inhibition or blocking this protein may enhance nerve growth in ALS. And so this is an exciting clinical trial that's going on now. So it's been well known that ALS, people with ALS, and we talk about it a lot in the clinic because we don't want people to lose weight, but people with ALS are often hypermetabolic. They're burning more calories. And this often results in weight loss. And it's thought that this energy depletion influences the development of disease and is associated with a worse prognosis. So people who don't lose weight or who are even a little bit overweight tend to do better, have a better prognosis, progress more slowly. So there have been a number of clinical trials evaluating high-calorie tube feeds. And there's one that's ongoing right now, and they're evaluating different strategies for high-calorie tube feeds, fat versus carbohydrate, so we can figure out what the best energy supply for people with ALS is and potentially slow the progression. There's also a trial that we're going to be participating in called olanzapine, which is a medication that promotes weight gain, and it also has neurotrophic effects. So it's thought that this may be of benefit in people with ALS. So Antisense technology is an amazing technology, and it goes along with a lot of the genetic technologies that are being developed, and there's other similar technologies. But if you remember from basic biology, to make a protein, you have DNA that gets transcribed into messenger RNA that gets translated into protein. And in many types of ALS, we know that these proteins are abnormal and tend to aggregate. So the approach is to try to keep from making those proteins. And so what's done is you synthesize a strand of oligonucleotides that can bind to the messenger RNA and make it not work so that you don't produce the protein. And so this is really powerful technology, and you can imagine that it may have a lot of benefit to particularly the familial types of ALS. And this has been looked at in SOD1 mice, where the antisense therapy, they were able to show that it actually does reduce the production of the protein and slow the progression of disease in the SOD1 mice with ALS. And there has been a recent phase 1 trial in humans that was a small trial, and it confirmed that this is a safe approach, but it was really too small to give much data about effectiveness. And so they're actively planning a phase 2 trial of this. And this antisense therapy is also being looked at in C9orf72, which I've said is the most common form of familial ALS and also underlies many cases of sporadic ALS. So this potentially could have much more widespread benefit. And so the abnormal gene for C9orf72 makes a messenger RNA that's really sticky and tends to clump together. And it's that clumping together that is toxic to the motor neurons. And so antisense therapy has been developed and used with this iPSC technology to look at ALS neurons in culture. And what they found is that using antisense reduced these abnormal RNA aggregates and seemed to decrease the susceptibility of the motor neurons to cell death. So this is also moving into human clinical trial. Just totally switch gears is a fascinating area called brain-computer interface. And there's a book that recently a physicist wrote about all the advances in brain development and the idea that sometime soon we're going to have chips that we can put into our brain that can extend our memory and function. But the brain-computer interface, so commonly with people with ALS, there's a concern about losing the ability to communicate because communication is based on motor skills. And so this is a type of technology that can bypass that. You can use brain waves to directly interact with computers to control computers and to use them for communication and for even operating robotic devices, mechanical devices. And so how this is done is that people are using biofeedback. People are taught to control some of their brain waves. And that, and then putting like an EEG, electroencephalogram cap on, those brain waves are converted into signals to the computer that communicate with the computer. And then you can use the computer for communication. And so there's been just a flurry of research trials looking at this in ALS patients. And it takes a lot of concentration, but what they found is that the cognitive skills necessary for this are well preserved in people with ALS to very advanced stages. So if you think back to one of the early slides I showed you with the list of all the potential factors that it's gone. Excuse me. With all the list of the potential factors that can affect motor neuron death, one of them was neuroinflammation. And one of the things that's well established is that many people with ALS have what's called a high inflammatory profile, so a bunch of markers of inflammation. And that the inflammation in ALS triggers macrophages to ingest both damaged but also healthy motor neurons. And macrophages are a type of blood cell that are sort of the garbage cans of the body, and they gobble up damaged cells or proteins. And so the excessive inflammation may in part be involved in progression of ALS. And so one of the things that's in clinical trial right now is tocilizumab, which is an antibody to the IL-6 receptor. And this is a part of the immune system that stimulates macrophages to attack neurons. And so it's hoped that by blocking this protein that the attack on the motor neurons will be blocked and there'll be better preservation of motor neurons. In SOD1 mice, it's been shown that this medication inhibits the inflammatory activation of the macrophages. There are many other therapies. Excuse me. There are many other therapies that are being developed to treat this inflammatory aspect. And this is my favorite, vitamin D. I think probably a lot of my patients have heard about how I've overdosed on vitamin D gummies. But vitamin D is thought to have a role in many neurologic diseases. And vitamin D is a potent steroid hormone with diverse biological functions, which include neuroprotective effects. And so there have been a few studies that look at the effects of vitamin D, and it's been shown that vitamin D deficiency, or marked vitamin D deficiency, was associated with a fourfold rate of increase in rate of decline and shorter life expectancy in people with ALS. When you look at motor neurons in petri dishes, it's also been shown that adding vitamin D seems to potentiate or increase the neuroprotective effect of certain growth factors. So vitamin D is an easy thing to do, and I'm tending to check that more in patients. The new RX diaphragmatic pacemaker system, I think many of you are familiar with, and it's a clinical trial that we're involved in at Providence. And this pacemaker was originally developed to treat people with spinal cord disease. And it's been being used with a very different idea in ALS. The idea in ALS is that using minimally invasive laparoscopic surgery, you implant electrodes into the diaphragm, which is the big breathing muscle of the body. And then those wires come out to the surface, and then several times a day, you attach those wires to a stimulator and stimulate the diaphragm. And the idea in ALS is that what you're doing is you're actually exercising the diaphragm and causing it to improve its strength and its reserve. So you're trying to build up the strength of the diaphragm so that as the disease progresses, there's more stamina and reserve. And this has been looked at in small clinical trials. So there's a study of about 82 or so patients wasn't a well-controlled study. So they didn't have controls, they just tried it in a number of patients. And what they found is that when you compared people who got the diaphragmatic pacemaker to similar patients at the same stage, that the people with the DPS lived about 16 months longer. And they were also able to show that by ultrasound, that the size of the diaphragm also got thicker, and the forced vital capacity, the breathing function, improved. And so what I would say is this is a very exciting trial that we're involved in, but despite the early promising data, it really needs a good trial because it is an invasive procedure, and so we need to really prove that it has benefit. So that's something we're moving along with. So exercise is another area of ALS that often comes up in discussions, and people always talk about their concern, or scientists do, that exercise will produce more oxidative free radicals or excitotoxins and put more stress on the motor neurons and cause them to die faster. But there's actually no data that this occurs. And in fact, it's thought that moderate aerobic exercise, such as stationary bicycling or exercising on a treadmill, may have the ability to keep muscles and nerves healthy longer. And there are a number of things that this moderate exercise does. It increases growth factor, it boosts energy supplies, removes damaged protein, reduces inflammation, and stabilizes the junction between the nerve and the muscle. So there was a study in Italy in 2011 that looked at about 40 patients with ALS and controls. And the ALS patients were exercised to moderate level of exercise, which was considered 70 to 75% of maximal heart rate. And what they found was that there seemed to be a slowing in the rate of decline of these patients and slowing of the rate of decline of the ALS functional rating scale. And I think most of you are familiar with the scale. It's a scale that measures multiple ALS functions over time. And so it seemed that that had a beneficial effect on patients. So there is now a trial that is looking at a much larger number of patients with different exercise interventions to look at progression. And it's long been known that if you take mice with ALS, so the SOD1 mice, and you take some mice and you have them run on the wheel their whole life, and you take some mice and have them be sedentary, the mice that run on the wheel live longer and do better. So this could be an important therapeutic intervention. And this is just one of my last slides that just to show you some of the things that are in the pipeline. There are many, many medications being developed that target all of those pathways that I mentioned early on. So reducing neuroinflammation, antioxidation, increased growth factors, neuroprotective, stem cells, and importantly, reducing protein aggregation. And so these are all things that are upcoming and maybe in trial sometime soon. So sort of in conclusion, this last few years has brought a marked increase in the understanding of the genetics and pathogenesis of ALS, which is translating into new models where we can develop new treatments. And there's also been a marked increase in the technology to develop these treatments, including the induced pluripotent stem cells. And there are a lot of exciting medications that are in the pipeline being developed now. And just a last slide that I just added because this data just came out. As you probably may know, Providence was involved in a clinical trial for tirasemtiv, which is a medication that works to increase the force of muscle contraction for every nerve impulse. And it's one of the first trials in a long time that has come back positive. What it showed that it actually seemed to improve strength and forced vital capacity in patients. But what they also found is that it had intolerable side effects, was not well tolerated. So what they're trying to decide now with this medication is how to move ahead with it, to alter it in a way that may be more tolerable to patients. So that's upcoming as well. And that's it. Sarah?
Yeah,
did
coming
you
up.
want to do
Yeah, so
questions at the end?
we're a little bit early, so if there are questions that people have kind of while Dr. Goslin's talk is fresh, we can go around and take a few of those, and then if you want to keep kind of maybe 3 or 4 questions now, if there are. Is that okay, Dr. Goslin?
Sure.
You up for? Okay, let's do a couple of questions now since we are a little bit early, and then we'll save the rest for our panel at the end. Yeah.
Yes.
If you're seeing that vitamin D may have a factor in this, are you finding that closer to the... Since you're seeing that vitamin D may have a factor in this, are you finding anything showing less of occurrence of ALS closer to the equator?
So to date, it seems that the distribution of ALS is pretty uniform throughout the world, with just some hot spots like Guam, for instance. But the data on ALS is very limited, and that's where the ALS Registry is coming in. So no, I mean, the obvious example is multiple sclerosis, which does have a increased incidence in areas where there's low vitamin D, in northern latitudes.
Dr. Goslin, tamoxifen showed up on one of your last slides, and I didn't catch it. Was there a suspected benefit or a detriment with tamoxifen?
Yeah, and I think that that's not one of the ones I talked about because I think the data for it is smaller. But I think there's a clinical trial with tamoxifen, and it arose from the experience of a single patient with ALS and breast cancer who progressed more slowly. And so from that point, they did some additional trial studies that seemed to suggest it might be helpful. I find that a little bit less powerful than some of the other things that are being looked at.
Have they identified any of the chemotoxic agents that are causing the inflammatory response in the macrophages?
So the answer is yes, that there's a whole profile that seems to be different in people with ALS, but it's quite complex, and there's just multiple, multiple cytokines.
Does it involve prostaglandin F2 alpha?
I'm not sure. It's such a complex area and there's many factors, so I don't know that it specifically involves that.
Let me bring the mic to you so everyone can hear back there.
Will your slides be available online?
I would imagine.
Yes, absolutely. We'll have slides and we'll have video recording online of the whole presentation, kind of with slides included. Yeah.
Can you give us the site so we can pull it?
Yes, we'll have to announce that, I think, a little bit later in terms of how we'll access it, but we are video recording in the back and have a plan to make it really widely accessible. So that will all be out there. Yes.
Was the moderate exercise trial, was that with the SOD1 patients, or was that widespread?
That was widespread, yeah. It wasn't any specific. And so I talk a lot about the research that's been done on the genetic forms of ALS, but a lot of that is felt to translate into sporadic as well.
Can you also say a little something about how the results and the information coming in from the registry may impact some of the work being done, and kind of the importance of it?
Yeah, well, so I mean, I think to date, there hasn't been, there's a lot of things that are thought about ALS but not generally proven, like the association with head trauma and athleticism and things like that. And the registry is just collecting a huge amount of data about that will lead us to a greater understanding of some of the environmental things in particular that cause ALS. So it's hugely important in terms of generating a better understanding.
Kelly, let me bring you the mic.
With the better understanding of the genetics of ALS, do you foresee a point in the future where everybody gets tested with their diagnosis
Yeah,
to kind of tailor the treatment?
That's a really good question, and I think I wanted to emphasize that without scaring people, that there is more of a genetic basis. And so I think there will be more of a role in the future of, and in fact, I'm doing that more and more in patients who are younger or slightly atypical, testing them as well. But because there's an underestimation of familial ALS, and some of the genes that cause ALS are found in the sporadic population, I think there's going to be more of a role, especially as treatments come about. These antisense or RNA interference is another form of antisense technology that are being used that are, I think, could be very, very effective at treating ALS. Yeah.
Thank you. Are there other questions that people want to ask now around the room? Yes, Marie.
I'm a total novice at this, but as you were going through this presentation, it seemed that the studies are basically being done to see, okay, now we have a patient with ALS, what can we do to slow the progression? And I'm wondering what kind of work other than the registry is being done to find out what tips the balance, what triggers this, since it appears that it can be many different backgrounds that people have and so forth. Is there work being done to try to figure out what is it that tips that balance, that one person may have the same gene pattern and they develop ALS and the next person does not?
So the answer is yes, but I think so much of the research is pointing at, is addressing that. So, some of the things I mentioned, like gene modifiers, that would make you more prone, and some of the genes that make you more susceptible to environmental toxins. So yes, it's being looked at extensively. It's just such a vast area that it is a huge part of the research that's going on. Yeah.
Okay, great. Thank you, Dr. Goslin. So we are running a few minutes. I'll come back up here. Ahead of schedule, which is fine. So we'll go ahead and take a break now for about 15 minutes, and we'll come together again. Feel free to go out and get some refreshments and use the restroom, and we'll join back for Dr. Beckman.